The benefit of 96 deep well plates is you have very high throughput, low sample volumes. It's compatible with automations and plate handling robots and liquid handlers. So it's easy to automate the whole workflow without inventing new equipment. And I've also heard of some pharma companies that have these automation, what they're called pods, where they do these kind of screening experiments performed almost entirely autonomously. But replicating bioreactor conditions in deep well plates is challenging due to the miniaturization of the liquid handling, the small volumes that you're working with, also miniaturizing the sensors from a bioreactor.
Welcome to the Smart Biotech Scientist. I'm your host, David Brolman, and today we're diving into the fascinating world of small-scale perfusion cell culture. If you've ever struggled with scaling down perfusion systems or wondered how to get more predictive data from your early-stage development work, this episode is your roadmap to success. I'm thrilled to introduce Tom Valentin, Group Leader at CSEM for Automated Sample Handling within the Life Sciences and Industry 4.0 business unit. CSEM is an internationally recognized Swiss innovation specialist. Tom's pioneering work in miniaturized perfusion systems is changing how biotech scientists like you approach process development. So grab your lab notebook and that fresh cup of coffee. In the next several minutes, you'll discover practical insights that could save you months of development time and transform your approach to perfusion culture. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brolman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Tom, to the Smart Biotech Scientist. It's good to have you on today.
Thanks for having me. Very excited.
Absolutely. Tom, share something that you believe about bioprocess development that most people disagree with.
As a non-bioprocess expert, I'll admit, based on a lot of the conversations that I've been having recently, I would say it's mostly related to continuous perfusion and whether some people believe it's the future or not for bioprocess development. I seem to think that it is, based on the conversations I've had, but I know there's also strong views in either camp.
Yeah, Smart Biotech Scientist, we would love to hear your take on this question because also as I'm speaking to a lot of people, I get different answers. So what do you think? Is perfusion or continuous processing the future or is it not? Tom, as both a mechanical and biomedical engineer, you bring a fascinating cross-disciplinary perspective to bioprocessing. So draw us into your story. What sparked your initial fascination with biotech and what pivotal moments led you to the work that you are currently doing?
So I find biotech to be a very interesting kind of marriage between advanced technology and biology, and get this kind of what we like to call a bioconvergence of technology and biology. And there's a very clear goal and use case, which I find very motivating as an engineer. It's an evolving field where the technical challenges are difficult, and that makes it exciting. Then for CSEM, I realized I was doing a postdoc and I realized that I wanted to do applied research that would be applied to the market very quickly. And I found out about CSEM. CSEM is a research and technology organization, the Swiss equivalent of Fraunhofer in Germany or CEA in France. So it's a partly government-funded research technology organization, and our goal is to support industry. So we do applied research and technology transfer, and that's kind of exactly what I was looking for.
Yeah, and I'm looking forward to our discussion because the perfusion system you're developing is definitely applied research, which and will make a big difference. But before we do that and dive really into the nuts and bolts of that microscale perfusion system, could you just briefly explain how 96 deep well plates and amber systems have transformed traditional fed batch process development? And also what limitations these technologies still face?
From a lot of the conversations that we've had, deep well plates have a lot of potential to serve as a high throughput and small scale model for both fed batch and perfusion, although for perfusion, it's not there yet. Doing it with fed batch, I think, has been around for a little while. The benefit of 96 deep well plates is you have very high throughput, low sample volumes. It's compatible with automations and plate handling robots and liquid handlers. So it's easy to automate the whole workflow without inventing new equipment. And I've also heard of some pharma companies that have these automation, what they're called pods, where they do these kind of screening experiments performed almost entirely autonomously. But replicating bioreactor conditions in deep well plates is challenging due to the miniaturization of the liquid handling, the small volumes that you're working with, also miniaturizing the sensors from a bioreactor. A lot of these bioreactors already have these sensors, but they're big. And shrinking those down to a scale that works in a 96 deep well plate, for example, with 300 microliters and 8 millimeter diameter, there's not very many solutions out there. I know there's a couple of advanced automated microbioreactor models for a fed batch. For example, the Amber 15, the 2MAG Bioreactor 48, the new Beckman-Coulter CSEM VT. You can have multiple feeds because they're kind of compatible with liquid handlers, so you can add different things in. You can test many different conditions, but in those, the sensing capabilities are often limited. You can do online pH and DO using these optodes. I know the CSEM has optical density for measuring cell density, but it's not viable cell density. That is done offline in the CSEM using a trypan blue imaging system. So with the limited sensing capabilities, it's hard to dynamically change your feed. So I think there's a lot of potential there, but there's also still a lot of limitations, both on liquid handling with the small volumes and then also the sensing. The highest throughput version, which is the CSEM VT, can do 96 samples, which is 24 deep well plates shaking in parallel. The others are limited to maximum 48, which is still a big increase in throughput, but we think there's potential to go even higher. And so we've had a number of discussions with companies in the biotech pharma space and even in the agriscience space, looking to automate these fed batch fermentation bioreactors in 96 deep well plates, then also doing perfusion. In some cases, they wanted to test 3 fully loaded stacked shaker incubators. I think almost 60 96 deep well plates in parallel, which is a massive screen. And doing that liquid handling is quite challenging.
Definitely, that's quite challenging. I've done a lot of 96 deep well plate fed batch experiments in my career. That can sometimes be a challenge when you do media optimization or some screening, but definitely when you're moving towards a perfusion approach, well, that adds a whole other layer of complexity to it. So many of you tuning in, probably you're familiar with the fed batch culture in some kind of small scale model, whether it's Amber or the other Beckman-Coulter, or maybe even in the 96 deep well plate. Now, you started developing a perfusion system in 96 deep well plate. What initially sparked your interest, and can you tell us a bit more about how your system works?
It all came a few years ago at conferences where we were demonstrating some of our technologies where we have microfluidic lids or fluidic kind of manifolds for well plates that we've developed, some of which will hopefully be launched to the market in June. And these enable medium exchange. We were able to do this for 6, 24, and 96, even 384 well plates. 384, just to be clear, is just pooling. That was for RNA sequencing. The 96 deep well plate one was for dosing experiments with organoids. The 24 well plate is just intermittent medium exchange. And then hopefully that will soon reach the market with Sullivan, a French startup, sometime this year. And then in other projects, we did continuous perfusion in a 6 well plate for regenerative medicine. And then we also did kind of continuous perfusion in an InnoSwiss project with Roche, where they have 40 microfluidic chips in a well plate format. And then we were able to continuously recirculate that with a 100 to 200 microliter working volume. So we had some of these demonstrators at conferences, and we were approached by companies who saw the potential in adapting this toward deep well plates for fermentation or continuous perfusion. And it was the high amount of industry interest that really sparked our interest. And we said, okay, we think that we have a impactful solution here. And when you pair that with an immediate market need, for me, that makes it very motivating and exciting. I think it's important to note that what we have in mind could also potentially be adapted for existing technologies like the Amber 15 2-mag or CSEM. So it's not just limited to 96 deep well plates, although that's kind of the use case that we have in mind at the moment. The core know-how for CSEM is really in this precision liquid handling. And so we can adapt those strategies to other methods as well.
Can you explain how you practically run one of these perfusion runs in the 96 deep well plates? What kind of equipment do you need to use, and what are the simple steps, like the overview of the steps that are involved?
For all of the use cases that we've seen, automated workflows are very important. And so having these kind of like plate hotels, shaker incubators, a robotic arm, a liquid handler, it's becoming more and more common, but we've seen that a lot. The most important things I would say are probably a liquid handler and a shaker incubator. Having the robot that moves things from one place to another helps speed it up and reduce the amount of manual labor that's needed. But I think the two most critical parts are the shaker incubator and the liquid handler, in addition to having a deep well plate. The concept that we have in mind would be you load the deep well plate with your cells, your media. It sits inside the shaker incubator. The high RPM and the large amplitude or throw enables good mixing, good DO exchange. And then you pair that with one of our liquid handling lids, and then you can get your continuous medium exchange through the lid. And that would run for several days, depending on the requirements of the process, what's the most important to each user. You can use different liquid handling strategies. So what we've done in some cases is using what we call pressure over liquid. So you provide air pressure on top of a reservoir of liquid, and that then pushes the liquid into each well, and you can do the opposite for withdrawing. In order to get that evenly distributed across the whole well plate, we use what we would call air restrictions. So that was a technology that we developed several years ago that is now part of Tecan's air restriction pipetter. So we can adapt that concept to a lid. The other strategy is not using pressure over liquid, but just injecting liquid. That is the strategy that we've been optimizing with Sullivan. There's pluses and minuses to both strategies. If you have several different media formulations that you want to test, then you need a separate tube going in for each media formulation if you're going to use liquid going in. If you have pressure over liquid, that allows you to then store all those media formulations in the lid in a bit of a kind of reservoir. And then you only provide air pressure going in, and then that media that's in each reservoir is then dispensed into the well. So the air restriction strategy would be kind of like a two-lid strategy, one the consumable reservoir, and then the other that does the distribution of the air pressure.
But in both options, or it's completely continuous, right? It's not a semi-continuous process.
The aim is for it to be continuous. So we can't necessarily at this time guarantee that it would be truly linear flow rate over 24 hours. It may be a bit pulsatile, but it would still always be flowing. The reason for that is the flow rates here are so low. So you want to go through 1.5 VVD and your working volume is 300 microliters. Exchanging that over 24 hours is a very low flow rate. And doing that with pressure over liquid is quite challenging. So it may be easier to give maybe air pulses rather than just continuous. And if you use the reservoir strategy, those reservoirs would need to be refilled, likely once a day. And so that means pausing the shaking for a little bit, taking it out, removing old media, adding new fresh media, then putting it back in the shaker incubator. For mammalian cell culture, I don't think that's as big of an issue to pause it for a little bit. For fermentation, it is a big problem because pausing for even a couple minutes can cause a big change.
Yeah, definitely. And how would then the sampling work? Is that in real time, or you still had to take the plate out and using the liquid handler to withdraw a certain volume to check the viable cell density, for instance?
So doing it with the liquid handler once a day is definitely the easiest. However, you can measure pH and DO in the well plate using pre-send sensors, for example. That kind of technology is already being used in the Amber, in the 2-mag, in the CSEM. So that's already existing technology. The optical density is used with the CSEM. It's also what's used in Merck's Mobius Breeze. What we've heard is it's less reliable for mammalian cell, better for microbial, but it allows at least a real-time measure of cell density, and then you can do viable cell density maybe once a day offline, and that might be enough of a compromise that's acceptable. We are very excited about one of our collaborators, another RTO. I'm sure many people know them, IMEC in Belgium and the Netherlands, are working on a technology that they call PatCube. And it can do glucose, lactate, pH, dissolved oxygen, and cell density, just total cell density, not viable cell density. They're developing this integrated thing for a bioreactor probe, but we think that there's a lot of potential there if the footprint can be shrunk a little bit, and they think it's possible to also do viable cell density. So if that could work, if that could be adapted to a smaller footprint that fits inside the well of a 96 or even a 24 deep well plate, then that would allow you to do real-time monitoring of all these things. And that could then enable these real-time changes. So if you're looking at fed batch in a deep well plate, you could monitor pH, DO, glucose, lactate, cell density, and then dynamically change your feeds.
Now, there's something else I'm trying to understand, because in the fed batch, it's not a problem because you sample and you do your analysis offline. But in perfusion, you have to handle the analytical part. Then, as we discussed, you have the media exchange part, which you manage to do with your technologies. But how do you keep the cells inside of your well? And also, how do you manage the bleed?
So for the cell retention, that's still a very challenging topic. We have not successfully yet demonstrated that in deep well plates, but we have in other projects. For diagnostics, we did it in a fluidic, where we then filtered blood and converted it to plasma, which was then delivered to a photonic chip, functionalized against proteins for breast and prostate cancer. In another one, we've done it for urine, which is actually quite challenging to filter. It's a very complex media. It's actually one of the more difficult ones to work with. And so we incorporated that into a point of care at home diagnostic. So you can take the urine sample, filter it, deliver it to your diagnostic, and get your result. We also have an ongoing project somewhat related to bioprocessing for a therapeutic. I wouldn't say it's a cell therapy, but it's based on cells. And in that, we've developed a kind of a customized TFF for filtration. So we have competences in that area, and we have a number of ideas for how to get cell retention in these well plates. But the first step right now is getting the continuous perfusion, because based on the list of priorities we've received, the most important is, can we get continuous perfusion?
And to get this to a point where companies can use it efficiently, what is the biggest challenge you're still facing?
I would say there's 3 big challenges that need to be overcome. The liquid handling for very low flow rates. I would say the liquid handling for the fed batch is probably the most challenging, because if you have a working volume of 300 microliters and you want to add a certain percentage over 10 to 12 days, it's not a full volume. And so adding such small amounts over such a long period of time is an extremely low flow rate. So that's very challenging. Continuous perfusion is a little bit easier, because if you go a little bit higher in your VVD, I think that's less impactful than too low. So higher flow rates are definitely easier to work with than lower flow rates. The next one is definitely the cell retention that we were just talking about. And then the other one is real-time biomonitoring. There is no system that we know of yet that can do viable cell density in a small enough footprint that could fit inside a deep well plate. We have the expertise, and I was brainstorming with some engineers about whether we could basically develop our own capacitance sensor, similar to like the Hamilton probe, for example, which seems to be the gold standard for real-time viable cell density. And making the sensor doesn't seem to be the hardest part. It's probably going to be more on the software and algorithm side. That could be doable, but it would be a big effort. Ideally, if someone else develops it and we can just get it off the shelf and integrate it into our fluidic lid, that would be the best. I would say there's one company, a small startup that we know in Paris called Inspect. One of our former colleagues is now the CTO there, and their strategy can monitor cell density now, but they're still testing it. I wouldn't say it's an entirely proven technology, and hopefully later on they can adapt it to viable cell density, but who knows yet. I'm not sure. In addition, there's also IMEC, who I think their system has a lot of potential as well. Solving this biomonitoring is definitely very important and still a big question mark.
And what are the most important parameters you need to monitor and to control in your small-scale perfusion systems?
The most important things that we've heard are pH, dissolved oxygen, and viable cell density. Now, people might be willing to compromise with cell density and doing viable cell density once a day. Glucose, lactate, other amino acids are also very important, but so far the most important ones are pH, DO, and cell density. And then the other critical process parameters are definitely also the liquid handling, getting your precision flow control. The shaking, I think the agitation is already pretty well established using those in shaker incubators.
And that wraps up part 1 of our conversation with Tom Valentin on small-scale perfusion cell culture. Isn't it fascinating how the right miniaturization approach can completely transform your process development strategy? Just imagine cutting weeks off your development timeline while getting more predictive data for your scale-up decisions. If you're facing challenges with your own perfusion setup or have questions about accelerating process development, I'd love to help. Book a free call with me to discover how you can fast-track your biotech development without those costly delays that keep you up at night. I'll personally help you apply these small-scale cell culture strategies to your specific biologics project. Just visit BrullmanConsulting.com/call to schedule a call with me. So join us for part 2, where Tom reveals the critical PAT strategies that make continuous processing at small scale not just possible, but transformative. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.